Um, okay, people. Shall we? Shall we get started? Okay, so we're in the last module of this first section, which we will probably finish sometime next week, and then you can start recapping, going over the material, give it a chance to process. If you have questions, let me know. We will have a review on the Wednesday before the exam, and the exam is on Friday, the 26th of February. So the Wednesday before will be the 24th. And if we are done with the material for section 1 early, which we are, then we'll just continue with the material for section 2. But the exam is only 1 through 6. So this last module is about solutes, solute movement in plants, and we're looking at a larger scale, and now we're also going to zoom in and look at a smaller scale, namely the membranes. So where we started was in the rhizosphere, and as you saw, plants have ways to make minerals more available to them. They can extrude protons, H+, which make cations more bioavailable, typically by replacing them from cation exchange sites on soil particles and humus particles. And then they're brought into solution so the plants can take them up. Also, plants can excrete chelating molecules that bind metals. And also, plants can have symbioses with microbes, both fungi and bacteria, in their rhizosphere, so the root zone, which can help them in many ways. And one of them is by producing chelators themselves, but also they can mediate other processes that make minerals more bioavailable. They may reduce them or oxidize them. Or they can also fight off other pathogenic microbes, and they can also help increase the root surface area. So plants have all these different ways to make minerals more bioavailable. Mineral bioavailability can be a limiting factor for plants' growth. And iron is the most difficult for plants to acquire because it is very unavailable. It's there, but it's stuck to the soil. It's not soluble. So plants have come up with basically 2 strategies called 1 and 2 to take up iron. And in both cases, chelators are involved. Iron just needs a chelator to be in solution. So that's a molecule that binds the iron and it keeps it dissolved. And for strategy 1, which you saw earlier, strategy 1, which is most plants, they use kind of non-defined chelators, which is just general phenolic compounds, which are compounds with rings in them. That are there in the rhizosphere. They're derived from plants, but also plants can make use of microbial chelators. And they bind the metal, bring them into solution. And then this chelated iron, which is in the form of 3+, that is the prevalent form of iron in oxic environments, like most soils, when there's oxygen present. That's bound iron 3+, that goes into solution attached to the chelator, can then move toward the roots, and it can find this membrane protein with enzyme activity called ferric reductase, and it can reduce the ferric iron to the ferrous iron, so iron 3+ to iron 2+. And the iron 2+ is more easily released from the chelator, so that's a reason why this reduction happens. And then once the iron 2+ is released from the chelator, it can be taken up by this other membrane protein, which you see down here, which allows the H+ to move through, and that is a process that requires energy indirectly. So the ferric reductase reduces iron 3+ to iron 2+. This is more easily released by the chelator, and then it can be taken up by a membrane protein. And you will see later, this is what's called a symporter, and we'll see later in this module what a symporter is. So this is a blow up of strategy 1. So this is what most plants use to acquire iron. And then strategy 2, this is where we left off last time. Those are the grasses, so that's a very specific group. And they have a special kind of chelator called phytosiderophores. And sidero means iron in Greek. Phore means to carry. Think of an amphora, something to carry things with. So siderophore is an iron-carrying molecule. And phyto means plant, so it's a plant-produced iron-carrying molecule. There are also bacterial siderophores. So these phytosiderophores are produced originally from methionine, which is an amino acid that's also in proteins. 3 methionines are combined to form one molecule of nicotianamine, and this can then be modified, can be tweaked a little bit, and then you get your phytosiderophore. Only grasses make phytosiderophores, but all plants have nicotianamine, and it plays a role in iron movement in the plant through the xylem and the phloem. So these phytosiderophores, or PS, are pumped out of the root into the apoplast, into the rhizosphere, and then they can go and get the iron 3+ from the soil particle and take it back to the root surface, where there is a transporter that can actually take up this siderophore iron complex into the cell. And once it's in the cell, the iron 3+ is reduced to iron 2+. It's released and then taken away by other chelators, and then the phytosiderophore is free again, and it can go back outside to find another iron molecule. So the reducing of the iron 3+ to the iron 2+ helps release it from the chelator again. And it's thought for kind of dangerous metals like iron and copper that they're always held by some molecule, some chelator. So even within the plant, wherever the iron or the copper is, there's always some chelator binding it, which can be just an acid or it can be a peptide. But because they're kind of dangerous, so they have to be chaperoned. And these molecules are also called chaperones. Unwanted interactions are avoided. So in the case of the phytosiderophores, they're oligopeptide chelators. They consist of 3 amino acids, basically, of methionine. And the group that methionine can use, that in general a lot of chelators can use, basically 2 groups combined iron. One is the thiol group, SH, and the other one is a carboxyl group, COO-. Those are 2 groups that tend to easily bind iron, and also other positively charged ions. So that's what you often see in biology. So the entire complex is taken up, it's reduced to iron 2+, released from the phytosiderophore, and then some other chelator takes it away, maybe nicotianamine. So that's the grasses. And grasses, the phytosiderophores of grasses are very good at capturing the iron, the sparse iron in the soil. So if you have competition for iron, which you often have, then grasses can often outcompete dicot plants and other plant species because they can very efficiently take up iron. And other plant species may make use of maybe microbes that can help them acquire iron. So this is blow up. So a little bit more about these plant symbioses with microbes. I already said that plants work together with microbes in their rhizosphere. The rhizosphere is the zone around 1 millimeter around the root. So the rhizosphere, so it's the sphere of influence of the rhizo, which is the root. And the microbial density in the rhizosphere is about 1,000-fold higher than in bulk soil, which means soil that is not under the influence of plants. So there are just way more microbes just right next to the roots. And that is because plants actually release a lot of carbon compounds from their roots. Not only because there's just turnover from root cells opening up, dying off, releasing their contents, so rhizodeposition, but there's also purposeful exudation of carbon molecules through the membrane of living cells. So plants do this on purpose, and it's been calculated, estimated, that it's at least 10% of the photosynthate that plants produce from photosynthesis, from fixing carbon, CO2, making sugars, that is released into the root zone to foster these interactions with these microbes, which are good for the plants in general. So the microbes are happy to live with these plants because they're fed with them. They're heterotrophic. They need to take up organic carbon like we do in order to live. And the plant is producing that organic carbon to the plant, to the microbes. That's true for both bacteria and fungi. So plants feed their microbes carbon, And in turn, microbes do something back. They help the plants take up minerals and water. They can ward off pathogenic microbes, so there can be biological warfare between the microbes. It's kind of similar to our gut. We benefit from the microbes in our gut, and we have good ones and bad ones, and the good ones can fight off the bad ones. We feed them all, and they give us something back in return. So they're warding off the pathogens and giving us nutrients, changing nutrients to more bioavailable forms. So that our microbes do stuff for us, and plant microbes do stuff for them. In addition to the rhizosphere microbes, there are also endophytic microbes, as it's called, which live inside the tissues of the plants, both in roots and shoots. And they can do many of these same things. So how do microbes help plants take up minerals? There are several different mechanisms. One is they can increase the root surface area, either by their own cells, their own tissues. If you think of fungi, they form these long threads called hyphae, which extend from the roots, and they can scavenge for water and minerals in all the little soil crevices and bring them to the root, deliver them to the plant as inorganic nutrients. So that's one way in which they can do that. Also, some microbes produce plant hormones, which induce root formation and root growth. So the microbes can make the plant itself grow more roots. So that can also be a way in which to increase the surface area of the plant itself by means of the microbes. So they can take up more minerals and water. Microbes can increase bioavailability, like if they could change a form that is, many of these nutrients can exist in different forms, as you saw, like protonated or less protonated, like H2PO4 versus HPO4. Or you have, for instance, you have iron 3 plus iron 2 plus, copper 2 plus copper plus. So you can have those kinds of reactions which can be affected by microbes. So bioavailability can be increased. And also, if you have siderophores, for instance, that are produced by microbes, that can increase bioavailability of iron. And some microbes can fix nitrogen. And the most well-known are the ones that live in nodules of legumes, the rhizobia. But there are other microbes that live also inside plants and that are not rhizobia, and they can also fix nitrogen for the plant. So what that means to fix nitrogen is to grab N2 gas from the atmosphere and turn it into ammonium and feed that to the plant. So they grab it out of thin air, so to speak. So these are all very important processes. And in agriculture and horticulture, the importance of microbes is increasingly recognized, and you can actually buy seeds that are already inoculated with microbes, and that gives you significantly better productivity than uninoculated seeds. So here are some examples of mycorrhizae. There are different forms. Some of them live kind of as a sheath around the roots of plants, and they're called the ectomycorrhizae, or ecto kind of means the outside. And these you see particularly for gymnosperms like pine trees. And you see that they do extend within the cortex with their hyphae, but it's relatively minor compared to the fungal biomass that is on the outside of the roots. And then you have the vascular arbuscular mycorrhizae, which you see here on the right. So they make these structures, vesicles and arbuscles, which really go within the cell wall, kind of within the plasma membrane perimeter, but there is a double membrane between the fungus and the plant. So they aren't really sticking into the cytosol. There is two membranes there, but it facilitates the exchange of molecules. This greatly increases the membrane contacts between the fungus and the plant. So they can exchange carbon going into the fungus and inorganic minerals and water going from the fungus to the plant. So these are VAM mycorrhizae. And at least 90% of the plant species live in symbiosis with mycorrhizae, and this may have facilitated the conquering of plants of land, of the continents, versus they came out of the sea originally and they were able to conquer the land where the climate is very dry. And these symbioses with fungi may have facilitated that process, and they're still very important for plant functioning. So at least 90% of plant species have them. And the ones that don't have mycorrhizae probably have other microbes that do these kinds of functions for them. And every plant tested so far has endophytes also. So this is something that's only recognized fairly recently, since we can have these non-culture based methods to detect microbes, where you could use molecular methods to do PCR, polymerase chain reaction, to see who is in there, who is in that plant, without the need to culture them. Because only about 1% of microbes are culturable in the first place, because people haven't figured out how to culture them, so you wouldn't see them if you were dependent on culture-based methods. But if you use molecular methods, you can detect them. So now there's a wealth of new information about the microbiome, as it's called, of organisms, including plants. It's a very interesting area. So that was a lot about the root zone. So what's going on here in the root zone? Very interesting happening place. The plants, the soils, and the microbes all working, interacting with each other. So let's have another look at what happens to these minerals. You already saw that the first step, of course, is the movement of the mineral into the symplast of the plant, which involves a membrane step, and that involves a membrane protein. So anything that moves, any mineral that moves across a membrane needs a membrane protein. And plants have tons of different membrane proteins, which are very specific for one particular solute at a time. So if our mineral is going to move on from the root to the shoot, it needs to enter the xylem of the root, which is apoplast. So that means there must be some export step out of the root cortex and into the xylem, which is apoplast. So that involves a second protein. And then we get translocation from the root xylem to the shoot xylem. And as you know, there are no membranes there, because this is dead cell walls left standing. There's a continuous water movement that is sucked up by transpiration, and minerals can just stay dissolved and go up with the flow. No membranes there. And then the last step would be the uptake from the xylem of the leaf into the symplast of the leaf, the mesophyll. So that involves another membrane step to get into those cells. And then depending on where our mineral ends up in the cell, it may need to pass additional membranes. Like to get into the vacuole, it would have to pass the tonoplast, the vacuolar membrane. Or if it has to go into the chloroplast, let's say it has to go to the chloroplast stroma, there are two membranes that surround the chloroplast to get in there. Or if it has to go to the thylakoid lumen, it has to pass three membranes. So there may be additional membranes to get to the final destination within a certain organelle. So those are the barriers that a mineral has to pass in order to get somewhere in the plant. So based on this, if you think about it, what is needed for mineral transport, for uptake into, or actually any transport across membranes? Typically it's an active process, not always. Many cases it's an active process, so ATP is needed to move it across a membrane. So to make ATP, often respiration needs to happen at sufficient rates. So if there's lack of oxygen, that may affect mineral uptake, because then you can't create enough ATP. And if you want to get minerals to the shoot, you have to have transpiration. So that depends on stomata being open, the plant having enough water to be able to have its stomata open and have transpiration, and then that water stream can carry the minerals up. So for translocation of minerals, we need the plant to have a good water status, to have enough water, not be dried out. That may affect mineral uptake. And in turn, mineral uptake affects water status, because you need to have the minerals in place in the roots to have that osmotic effect to draw the water in. So you have to have the minerals there as solutes to lower that solute potential to get the water in. So it's kind of a minerals affect water uptake, and then water status is needed to drive mineral movement in plants. So to look a little bit more close up at that step of first the uptake into the symplast, which may happen at the root here, or it may happen at the very latest here at the endodermis. So at that point, the mineral will have to pass a membrane into the symplast, the interior of the root cells. And then after that, the second step would be, once it's in the endodermis, if it wants to get into the xylem, then it would have to pass out of the symplast, as you see here, and into the apoplast of the xylem. So you see it goes from blue to pink, where pink represents apoplast, so outside of the plasma membrane, and blue is within the symplast. So there must be a membrane step every time we switch color. So if we go from apo to symplast, there's a membrane there to get into the cell. And if we go from blue to pink again, there's another membrane step to get out of the cell. So the reason why every mineral needs to go into the symplast before it can go into the apoplast of the xylem is because the endodermis, this orange cell layer, has an impermeable cell wall. So things cannot freely diffuse through there. No water, no minerals. So that's the function, is to keep the good minerals in and be able to accumulate them without leaking them, and to keep unwanted or excess minerals out. So we have ions passing through a membrane for the translocation, as it's called, from the root to the shoot. It has to enter the xylem in the root, and for that the ions have to pass a membrane, and this involves ATP, actively, directly or indirectly. So it's some form of active transport, and we'll get back to what active means when we zoom in on these different transport mechanisms later in this module. So in the xylem, there is just movement up with the water flow, which only happens when the stomata are open and the plant is transpiring. Then it's sucking on this water column. And adhesion and cohesion help of the water molecules. And the membranes are just the minerals are just dissolved and they just go with the water. No membranes. Driven by transpiration. And then for the uptake into the mesophyll cells, the ions have to pass a membrane to get into the symplast. And that is again an active process usually, so it needs some ATP to make that happen. So now, so far what you saw was the mineral started in the soil water solution. It moved through the root symplast via the xylem apoplast into the leaf symplast. That isn't necessarily the final destination of our mineral. I already said it might move further through more membranes to get into a certain organelle. But also at some point in the life of the plant, it may want to remobilize our mineral to other parts of the plant. For instance, if this is an annual plant, then it will survive the winter as a seed, and so it will at some point start to remobilize minerals from its leaves as the plant is starting to senesce, and it will try to put it in the fruit and in the seed. And if it's a perennial plant, but if it's deciduous, then it will try to remobilize the minerals from the leaves and bring them back to the root or in the stem, and then the rest of the leaves are going to drop off. So remobilization is something that is part of normal life of a plant, and that involves the loading of the mineral into the phloem. So that involves actually membrane transport again. Typically the phloem has no plasmodesmata connections with the surrounding tissue, so that means there are two membrane steps to be taken for things to go from adjacent cells of phloem into that phloem, because they're both living cells. Phloem is living and the adjacent cells around it is living. So there are two membranes, no plasmodesmata usually. There are exceptions. So that means two membrane steps, and that gives again the plant control. So there are again transporters involved, and energy is needed to remobilize those elements. And as you already saw in the previous module, not every element can be remobilized equally easily. As you see when you get deficiency symptoms, you see it in the younger or in the older leaves. So if you see it in the older leaves, the plant, that mineral can be remobilized easily, because then plants will remobilize it if it can, to young leaves. And if you see deficiency symptoms in the young leaves, that is an indication that the element in question that's deficient is not very mobile in the phloem. So that was a little bit about just whole plant transport processes that happen via the xylem and the phloem and the membrane steps involved. A little bit about regulation. So I mentioned earlier that a plant must be able to sense how much it has of every element in every part of so every organ and every tissue and every organelle even. So it must have sensing mechanisms, and then when it is getting deficient, so when it gets into that deficiency zone, it has to ramp up its recruitment of that element, and if it is in the adequate zone, it can ramp down the further recruitment of that component, because you could say it would be just a waste and it might even get toxic. So that's homeostasis, as it's called. So you have homeostasis at the cell level, but you also have homeostasis at the whole plant level. So the shoot, for instance, can send signals down to the roots to tell the roots, I need more of a certain element, or I have enough of a certain element. And there's still relatively little known about how this works. There's just for a few elements this has been found how that works. So what has been found is there can be local signaling at the root cell level. If you have simply the solute itself accumulating, then that can down-regulate the transporter that takes it up. So that's very direct regulation, so that the mineral itself may locally inhibit further uptake when it starts to accumulate, kind of feedback inhibition. You can also have that there is a downstream processing molecule, like if you have sulfur that is assimilated into amino acids, then one of those downstream molecules can act as a signal to downregulate further uptake of sulfate when it starts to accumulate. So a downstream molecule may act as a signal. Yeah, question? Okay, so why would you see toxicity if plants are so good at stopping uptake? Yeah, they will try to do this, and they will keep doing this to a certain point, but if the plant is really overwhelmed with a certain element, then you can break through this, you could call it the defense of excluding, and then the plant is so overwhelmed that it will start to suffer toxicity anyway. Yeah. So a possible signal molecule could be a downstream processing product that is derived from the mineral in question, and in some cases it can actually be small RNAs which can travel in the phloem, and they have a signaling function. So these can be produced in the shoot, travel in the phloem down to the root, and then they can then regulate the expression, the production of the transporter. So there's just some things known about how this is regulated, but it's still sparse, but it gives us an idea of regulation mechanisms, but there's still a lot to be discovered. So the transporter protein may be affected by these various signals, the minerals or a downstream metabolite or a small RNA. They may downregulate the production of the transporter, so just at the transcript level or the translation level. Or in some cases the transporter is actually taken out of the membrane and stored in a vesicle, and then when it's needed again it can be placed back in the membrane. That's another mechanism. Or it could be activated, inactivated via phosphorylation. So those are just some modes of regulation. Or there could be interaction between proteins, protein-protein interaction that down-regulates the activity of a certain transporter. So there are different modes of regulation. Complex but interesting. So that was all kind of whole plants, needs, movements of minerals. Any questions about that before we delve into membrane transport mechanisms? Okay. So all these minerals need to move across membranes, so let's have a look at how that works. And while we're looking at that, these things that you're going to see are not just for minerals, for inorganics, but they are general membrane transport mechanisms. So they also are true for amino acids, sugars, you name it, anything. Keep that in mind. So this is pretty basic stuff, and most of it is not even just plants, but all organisms have these similar mechanisms. So just a reminder, the membranes consist of phospholipid bilayers, which are these balls with two tails. So a hydrophilic head and two hydrophobic tails. Exception, membranes of chloroplasts can have sulfolipids instead of phospholipids, which may reflect their endosymbiotic origin as a bacterium. But most membranes phospholipids, and that's about 50% by weight of every membrane, and the other 50% is proteins. And these proteins include many, many different transporters, which each transport a particular substrate. They're very specific. And they have certain properties. Here we see an example of a protein. And this ribbon that you see is an amino acid chain. And it can fold spontaneously in these secondary structures, like these alpha helices, which form membrane-spanning domains. And you can also have folds called beta sheets So based just on the properties of the side groups of all the amino acids that together make up this chain of protein, this protein kind of spontaneously folds in this conformation, and then it's in that form only is it functional. In this case, you see that this protein forms a pore. So some of these membrane proteins that facilitate transport are pores that can allow things to move through. And this is called an integral membrane protein if it spans the entire membrane. So it sticks out on both ends. You also have peripheral membrane proteins, which are just attached on one side or the other. But the real transporters are integral membrane proteins. They really have to be able to span the entire thickness of the protein, of the membrane. So one important concept in transport and also in general in reactions is passive versus active. Passive transport means that the process does not require any energy. And when we're talking about energy in cells, typically we're talking about ATP. That's the currency of energy. So passive transport does not require ATP to happen. And you could say no energy is needed because it goes from high to low concentration. You could also reason the other way and say, because no energy is put into this reaction, it can only go from high to low concentration. You cannot pump something or bring something up the mountain without putting any energy into it, either sweat or gasoline. But getting things down the mountain is easy. You just let it go, and there it goes. It spontaneously goes. So passive transport goes from high to low concentration, requires no energy. And you see here as an example, we have here the two A's. In one case, a molecule just diffuses from high to low concentration in between the phospholipid molecules. That's pretty rare that molecules can do this. And in the case of minerals, if they're charged, this is not a feasible option. Then they have to make use of a membrane protein. And then a passive transporter may look like this. A channel, which just has a hole in the center, and this forms a hydrophilic pore through which a mineral can pass. So that is an example of passive transport. In an active transport, you see here as B, it requires energy, and it uses that energy to move things across the membrane up the concentration gradient, so from low to high concentration. And these are also called pumps. So they pump things up the concentration gradient. They need ATP. And they may need the ATP directly or indirectly. And you will see more about that in a minute. So active transport requires ATP, requires energy. Goes from low to high concentration, typically from low to high. And often cells have two different transporters in a membrane, and if they have to go up the concentration gradient, they will use their active transporter. But if they can get away with using a passive transporter, they will. Because why would you pay for something if you can get it for free? So passive transport goes from high to low concentration. It's driven by diffusion. Diffusion is simply molecular motion. If all molecules are moving all the time, you wouldn't say it while we're sitting here, but all our molecules are moving all the time, bouncing around. Higher the temperature, the bigger the motion. And the probability is just higher. If the concentration is higher in one place than another, the probability will just tell if everybody's in motion that after a certain time, things have moved from high to low than from low to high. So this is just a general physical law. Things go from high to low concentration, and the process is called diffusion. And that drives the passive movements of molecules, also across membranes. Requires no energy, high to low concentration. And if it just goes through the bilayer by itself without the need of a protein, it's not specific, and nobody's helping the solute. It's called simple diffusion. It can also go by means of a channel protein, in which case it is somewhat specific, a little bit specific, but it cannot be highly specific. So the only specificity that such a protein can have is the size of the pore. You have to be small enough in order to go through, so that is some sort of exclusivity. And the other thing is, if you're charged, then such a channel can often only allow, for instance, positively charged ions through, because it has negative charges on the inside of the channel, so negative solutes are going to be repelled, so they're not going to go through. So there's a little bit of specificity, but it's not highly specific. There's also a kind of membrane transporter which is not shown here, but I'll show it later. It's called a carrier, and it is a passive transporter, doesn't need energy to do its job. It only goes from high to low concentration, but it has more specificity because it actually binds the solute. And that is also called facilitated diffusion. So it's diffusion, it goes only from high to low concentration, and it's facilitated by this carrier. So here we have a model, a very simple model of how such a carrier might work. It is a specific transporter, meaning that it has a binding site. So it has a binding site that the solute binds to, and then somehow, without spending energy, this protein can move the solute across the membrane down a concentration gradient, as you see, because there's a higher solute concentration here on top than here below. So people have come up with a model, well, maybe it bulbs kind of. If you have ping pong balls floating in a dish pan, they can just roll around on top of the water, and that doesn't cost much energy. So maybe it's something like that. The carrier may be able to flip-flop, as it's called, in the membrane. So then the binding site is either exposed to one side of the membrane or the other, but the protein doesn't undergo a conformation change, which would require energy. So these simple diffusion you see here, the higher the external concentration, the faster the diffusion goes. And this is typically the kind of uptake rates that you see for simple diffusion and for carriers. It's not saturable, so it has an unlimited capacity. It's not saturable because there's no binding site on the channel. But the carriers, they facilitate the diffusion, as you see here. At low concentrations, they can make it happen faster than a channel, but it saturates. So at some point, all the binding sites have a substrate bound, have a solute bound already. So there are these yellow balls in every binding site, and then you can give more substrate, but it's not going to help because the carriers are all busy. So this is an indication, if you see this kind of uptake rate curve, indication that the protein has a binding site, and that also gives it more specificity, because a binding site can be specific, like a lock and a key. So that's a carrier. That's called facilitated diffusion. So we're still in the passive transport mechanisms. Here we are going back to the channel. So channel is basically a hole in the membrane, a pore. And like I said, they can't be too specific. It's just a hole, so I guess you have to be small enough to go through the hole. And then there can be some charge specificity, like only if you're positively charged can you enter in this channel, because the interior is negative. So even though it's a hole, you might think, well, aren't these holes leaky? How can you keep things in or out? There is actually a mechanism by which channels can get closed and opened, and this is called gating. So a gated channel it really looks like a little gate, like you may have in a garden, like a little flap of protein sequence, which can just be moved, opened and closed. And this can, for instance, happen by a change in charge across the membrane, called membrane potential, that may suddenly cause this gate to shut closed or to open back up. And so this, for instance, you can imagine that if this is a potassium channel, here we have our potassium, K+, that if this little flap of protein is positively charged and it slaps closed and it's positively charged, the potassium cannot get in anymore. So this may be a way, if you have, as you know, potassium and chloride are osmolytes. So if plants want to move water, let's say into guard cells, then they want to move potassium and chloride via channels quickly first, and then water follows by osmosis, but afterward you want to close these channels, otherwise potassium and the chloride would flow right back out. So that can be closed, and you see there's no binding site, so the potassium can just flow through it, it's never bound. That gives the channels a very high capacity, and it makes them super equipped to do really fast movement of solutes. Like if you consider a Venus flytrap, snap, that goes pretty fast. And that's because channels are used to move potassium and chloride, and then to move water by osmosis, again, channels, aquaporins, remember those guys? So that's a bunch of channels which give this very quick movement of K+, Cl-, and then water. And you see how fast it happens because you can see it happen in front of your eyes in a matter of seconds. So if something really fast needs to happen, channels are the best mode to get things across membranes. They have very high capacity, and that's because they don't bind their substrate, because the process of binding and releasing, that takes time. So that makes them more specific, but less fast. Yeah?
So in a case like that, would the plant keep a lot of those elements right outside of the gate? So then that's a high concentration. So when the gate does open, and they can move fast in, and then when they don't need it, then it's high concentration inside of the cell.
Yeah, typically the K plus and Cl minus just shuttles back and forth between two compartments that are separated by a channel. And then they can be moved by opening this channel under certain conditions, so that the opening and closing is triggered by something, like a light, sun coming up or something. So that was all passive transport down the concentration gradient. So we had simple diffusion without any protein, which is, you could say it's negligible for minerals because they're typically charged. Then there are channels and there are carriers, passive transporters. That's passive. Now there's active transport, which is basically two kinds of transporters, which are called primary active and secondary active. Either way, they need ATP somehow, and that is because these transporters function in moving things against the concentration gradient from low to high concentration. There's always a membrane protein in this case, and that's often called a pump. Particularly, it's called a pump if it's primary active, which means it actually needs to physically interact with ATP and get energized by ATP to pump its substrate. Active transport is always specific. There's always a binding site. So the proteins involved always have a binding site. Molecules are pumped across the membrane against their concentration gradient, meaning from low to high concentration. And the results can be quite impressive. For instance, if you would measure the concentration of a certain element in the tissue of the plant and you compare it to the concentration in the soil, you may find a difference of 1,000-fold or more. And that's also called the bioconcentration factor, or BCF. Bioconcentration factor is the concentration inside the plant divided by the concentration outside the plant. So it's the degree to which the plant concentrates a certain element. So it's pretty impressive that a plant can concentrate something across the membrane 1,000-fold. That's a pretty strong pump and a pretty non-leaky storage mechanism. So here we see the passive transport again, so simple diffusion, channels, and carriers. Where channels are also considered simple diffusion and carriers are mode of transport by carriers is also called facilitated diffusion. That's all passive. Diffusion is passive. So now active, you see here a pump. It pumps something up the concentration gradient, which is not really clear by showing the solutes, but that's shown here by means of this purple kind of triangular arrow. This represents the gradient. So our solute here goes against the gradient. And it says here electrochemical potential gradient. I'll come to that in a little bit. But that is the sum of the concentration difference and the charge difference across the membrane, because those are the two factors that determine to what extent a charged solute will spontaneously move across this membrane. It will spontaneously tend to go down its concentration gradient, but also it will spontaneously move to the side of the membrane that has an opposite overall charge. So if the ion itself, like K plus, is positively charged, and there's a charge difference across the membrane called the membrane potential, then our positively charged ion will spontaneously tend to diffuse to the negative side of the membrane. So the combined force of charge difference across the membrane and concentration difference we call the electrochemical gradient. So active transporters are needed when things have to move against this natural force. So primary active transporters require energy directly. The protein in the membrane physically interacts with ATP. It receives a phosphate group from ATP, which energizes that protein in the membrane, and that allows it to change its conformation, that means its shape, and that this change of shape makes it possible to pump the solute across the membrane. And after it has released that solute, it falls back into the original conformation. And that means that another ATP molecule is needed to phosphorylate it again, change the shape again, and then a second solute molecule can be pumped. So an ATP molecule is needed for every molecule of solutes. And the ATP is needed to bring about this change in shape of the protein. Primary active. Secondary active, we'll see in a minute how that works. Any questions so far about this passive versus active transport? Okay. So you may envision active transport, for instance, like this. This M plus could be a metal, for instance, like this is how copper gets across membranes, how other metals may get across membranes. So you have your M plus, your metal. Here's the binding site. This is a primary active transporter. So the metal can bind to the binding site. Now an ATP molecule is needed to stick a phosphate group, kind of you could say it's in an activator, as an activator function, to this allosteric site. Now the phosphate group is in place. This allows our membrane protein to change conformation. And you see how it changes from this conformation here to this conformation. It's different. So the energy is needed to make that happen. And now you see that the binding site is now exposed to the other side of the membrane. And now the metal can be released. So this conformation may also allow for this metal to be released. And then after the metal is released, it falls back into the original conformation. And then the phosphate group is lost. And then a new ATP is needed so this is a cycle, goes right back. So this can keep going and going and going. But for every M plus to go through that membrane, you need another ATP molecule. Any questions? So the most important primary active transporter is the proton pump. You heard already that plants acidify their apoplast, including the rhizosphere, by pumping out H+. And this protein complex that does this is called the proton pump. It's also called ATPase because it hydrolyzes ATP in the process. So it breaks down ATP and ADP and P. And in that sense, it's similar to other primary active transporters. So the ATP gives a phosphate group to the proton pump, and that then allows it to do its job and pump H+ across the membrane and against the electrochemical gradient and against the concentration gradient. And that acidifies the apoplast, which typically is something like pH 5 or 5.5, whereas the cytoplasm is typically pH 7.4 or something. So this pH difference is created by the proton pump. And the proton pump has two very important effects, which are super important for the movement of other solutes across the membrane, as you will see by secondary active transporters. So it creates a proton gradient, which facilitates the uptake of other elements, and it's a big contributor to the membrane potential. So what that is the unequal distribution of charges across the membrane. H+, positively charged, if you keep pumping that out, this creates an overall positive charge in the apoplast relative to the cytoplasm. See? And this is actually, you can measure it as a voltage, and this may be something like 120 millivolts, or even in some membranes, membrane potential can be 200 millivolts or something. And just to give you an idea, in the socket, it's 110 volts. So that's pretty high for such a small cell. So this membrane potential is potential energy, like the socket is potential energy. You can plug into it. So similarly, other transport processes can plug into this potential energy of this membrane potential. So we'll see next time how the H+ gradient and the membrane potential facilitate the movement of other solutes. Have a great weekend.